Good morning. So I'll be talking about the complement system. The complement system is a crucial component for the innate immunity because it consists over 30 proteins and basically it works in a cascade mechanism to eliminate the pathogens that enter our body. It bridges the innate and adaptive immunity, facilitating processes like opsonization, inflammation, and cell lysis. So basically, it is a group of plasma proteins which are also called as complement proteins, and they are produced in liver and they help to destroy the pathogens. So the cascade mechanism is basically what we will see in this seminar. This is the overview. You can go through this. So firstly, the components and activation, how it occurs. The complement system can be activated via three distinct pathways: the classical, lectin, and alternative pathways. So each pathway converges to a central component C3, leading to a cascade that results in pathogen elimination. So let's see how these pathways work. The classical pathway, it is initiated by antigen-antibody complexes as seen here, and specifically IgG or IgM binding to the complementary C1q. This activation leads to a cascade involving C1, various cascades of C proteins, and ultimately it forms the C3 with the help of C3 convertase enzyme. There is lectin pathway. It is triggered by mannose binding lectin or phagocytic binding to specific carbohydrate patterns on microbial surfaces. So this pathway will activate mannose binding lectin associated serine proteinases, which then cleave C4 and C2 and forming the same C3 convertase as in the classical pathway. Then there is alternative pathway, and it is like always active, and it works mostly spontaneously and on the microbial surfaces without the need of antibodies. It involves the hydrolysis of C3, leading to the formation of C3 convertase enzyme, which specifically amplifies the response. There is one more called as the convergence and terminal pathway. All pathways lead to the formation of C5 convertase, which cleaves the C5 into C5a and C5b. C5b initiates the assembly of membrane attack complex, compromising the C6, C7, C8 and multiple C9 molecules, accumulating in pathogen lysis. So here is a brief overview. We can see the classical pathway forms the antibodies, whereas the lectin pathway will form mannose binding lectin, and alternative pathway will do C3 hydrolysis to basically form the C3. They all converge to form C3 and C3A and attack membrane attack complex. So the C3A formation will lead to chemotraction, and membrane attack complex will lead to opsonization. So we'll see what are the biological consequences of the complement system. So activation of the complement system will result in several effector functions enhancing the immune response. Firstly, there is opsonization. It is the binding of C3b to the pathogen surface, making them marking them for phagocytosis by immune cells equipped with complement receptors. So the opsonization is basically, it is a marker for the natural killer cells or other phagocytic cells to attack to the specific pathogens. It also causes inflammation. It is anaphylactoxins C3A and C5A increases vascular permeability and attract immune cells to the site of infection, promoting inflammation. Then there is cell lysis. It is the membrane attack complex that form pores in the membranes of the target cell, leading to osmolytic imbalance and eventually the death of the cell. Also, there is immune clearance, like complement components that will aid in the clearance of the immune complexes and apoptotic cells, preventing autoimmunity. So it is also managing the autoimmunity prevention. Now we'll see how complement regulation occurs. So to prevent damage to the host tissues, the complement system is tightly regulated and by various complement control proteins which are present in the blood plasma and host cell membrane. So the key regulators, there are 4 key regulators. These regulators ensure that the complement activation is targeted and does not harm the host tissues. So there is decay accelerating factor DAF that disrupts the formation of C3 convertase and prevents the complement system. There is C1 inhibitor. These inhibit the activation of C1R and C1S in the classical pathway. Then there is F and H regulator. Factor H and factor I regulator. These down regulates the alternative pathway by inactivating C3B, and there is protectin CD59. So this prevents the formation of membrane attack complex on the host cells. So this is how the host cells are protected from their own complement system. We will now see how the complement system deficiency can cause various diseases in the humans. So deficiencies in the complement system components can lead to the increase in susceptibility to infections and autoimmune diseases. So here is a table to summarize it. Basically, if there is C1, C2, C4, so it can cause immune complex diseases such as systematic lupus erythematosus, or if there is C5, C9, it can cause increased susceptibility to Neisseria. And there is C1 inhibitor, it can cause hereditary autoimmunogenic edema. So the classical pathway deficiencies, the deficiencies in the C1, C2 and C4 are associated with increased risk of autoimmune diseases such as SLE. Then the C3 deficiencies, it can lead to the recurrent pyogenic infections due to impaired opsonization and membrane attack complex formation. Then the terminal pathway deficiencies, it is C5 to C9. This results in the increased susceptibility to certain bacteria such as Neisseria, particularly causing meningococcal disease. Then regulatory protein deficiencies. So C1INH deficiency causes hereditary angioedema characterized by recurrent swelling episodes. Properdin deficiency, it is an X-linked disorder that leads to increased risk of Neisseria infections. So these are some of the diseases that can be caused due to the deficiencies in the complement system. What can be the diagnostic uses of this system? So assessing the complementary system can activate complementary system activity is vital in diagnosing and monitoring various immunological disorders. So here are some examples. There is AH50SA. So this assesses the alternative pathways functionality. Then there is C3, C4 levels. These levels are evaluated in the conditions like SLE. So decreased levels will indicate the complement system is hampered, and it can be a marker for this disease. Then flow cytometry can be used to detect deficiencies in regulatory proteins such as CD55, CD59, aiding in the diagnosing conditions like paroxysmal nocturnal hemoglobinuria. Then some clinical applications can also be used, such as complement systems. The complement components can serve as a biomarker in diseases such as multiple sclerosis, elevated C3, C4A levels correlated with the disease activity. Then CH50 assay, it measures the functionality activity of the classical pathway. So all these assays can be used using the blood which can serve as a biomarker and can be used for diagnostic purposes. So there are a few recent research which I have mentioned here. Complement as a therapeutic target. So research is ongoing into complement inhibitors such as potential treatments for diseases like age-related macular degeneration and atypical hemolytic uremia syndrome. There is complement in autoimmunity. So studies have shown that deficiencies in the early complement components are linked to development of the autoimmune diseases due to indirect clearance of apoptotic cells. Also, there are some research who from over the last decade have shown that the complement proteins of the classical complement pathway have an important role in the synaptic forming in the brain during the early development. So the complement system can be summarized with this pathway. There is the antigen-antibody binding, and the fragments are converted into the C3 using the C3 convertase enzyme. And alternatively, there is hydrolysis which can also form the C3 enzyme, and these then attach to the pathogens and basically helps the immune system to mark the pathogen and attack to this. So this is a very important part of our immune system. Thank you.